Hey everyone, in this video I want to briefly introduce transposable elements. Now these are genetic elements that can spread throughout a genome and they can increase in number. Some people or researchers consider them selfish. Others say they're slightly like viruses that don't have or can't exist outside of the cell. But let's just talk about what they are. And we're going to, in this video, talk about one, maybe the simplest form of a transposable element called an insertion sequence. But first, I think it's probably easiest to introduce the different classes. So I guess the simplest classification system for transposable elements divides them into two classes. So we have the class 1 elements, which spread by a copy and paste mechanism. And these involve an enzyme that we've seen a couple times so far in this course. That's reverse transcriptase. And the class 2 transposable elements, these spread by a cut and paste mechanism. And these have a transposase. So one of the key enzymes in moving or helping a class 2 transposable element move to a new part of the genome is it's called a transposase. And in this video, we're gonna take a look at the cut and paste mechanism, and in future videos we will look at the copy and paste mechanism. Now, as promised, I am going to talk about a very simple type of class 2 element. So one example of a class 2 element, or one, I should say, a group of class 2 element are known as insertion sequences, and these are typically found in prokaryotic genomes, but there are some eukaryotes that have insertion sequence like class 2 elements. Essentially, these are, it might help if I just diagram them, right? So they're class 2 elements and they are relatively short, to be considered an insertion sequence. It will be relatively short. So the range, I think the current known range for these is between 700 base pairs and 2,500 base pairs. So a pretty short sequence for an insertion sequence, and these can also be called IS elements. So IS for insertion sequence and IS element. Okay, so here's an IS element. Let's say in part of a chromosome, let's say chromosome of E. coli. It's a circular chromosome, but I'm only diagramming part of the chromosome, and I'm indicating the size range to be between 700 and 2,500 base pairs. Now, insertion sequences have on the ends these things called TIRs. So two TIRs, TIR, and TIR stands for terminal inverted repeat. And okay, so what is a terminal inverted repeat? Some of you probably know or can probably guess what I mean by that, but let me just come up with an imaginary terminal inverted repeat. So let's say at the end of this IS element over here, we have an AAA GGG in the top strand, and then 3 T's and 2 C's down here. Now, if this is on one end, and this is a terminal inverted repeat, or one member of a terminal inverted repeat, the other side is going to have the same sequence, just inverted. So the AAA is going to be down here, and TTT over here, and then, oh, did I say that? I just messed that up, didn't I? Sorry about that. No, that's not going to happen. That would be really weird if it worked like that. So TTT up here, CCC, and GGG. Okay, so this one is going that way, the other one is going that way. Okay, so we have our inverted repeat. Now, and that's just one example, and they could be longer, and you know, maybe there's not an IS element in the world that actually uses this sequence. But this is, I just wanted to show you the inverted repeat, inverted nature of the TIRs. So now outside of the TIRs, there is going to be something else. And these are going to be called TSDs. So one TSD over here and another TSD over there. And that is, that stands for target site duplication. So it's something about the way IS elements insert into a target site in a genome. The particular way they insert causes a duplication of the target site. So we get a target site duplication. Now the lengths and the sequences of the TIRs and the lengths of the target site duplications are specific to different IS elements. And they're going to be related to the transposase, which is encoded by the insertion sequence. So the IS elements are relatively short, and they usually only encode one gene, and that would be for the transposase, but some have some encode another protein, which would be a regulatory protein. So we are not going to talk about those that, you know, also encode a regulatory protein. We'll just focus on, you know, a very simple case of an IS element that encodes one gene, just a transposase, between the TIRs and the TSDs. And that transposase, you know, it is going to be specific for sequences found in the TIRs, so it will recognize those and it will bind those. And then when it's processing the TIRs, when it's cutting the DNA to release the IS element from the DNA to help it move to a new location in the genome, the way it cuts the DNA, that's going to lead to the target site duplication of specific lengths. I'm going to show you exactly how that process works in the next diagrams. Okay, I think that's enough for the introduction. Okay, and I cheated a little bit here. I've already created some diagrams and put some color into them. So let's imagine that this is just a part of a bacterial chromosome, say E. coli, and here's the insertion sequence. Maybe it's about 2,000 base pairs long. Here are the TIRs, and here are the here's a TSD, same thing over here. So, okay, how does this move to a new location? Okay, so insertion sequences, IS elements, that's what they do. They move to new locations of the genome. So what would happen was, you know, whatever the transposase that is in here would be transcribed by RNA polymerase. So I have transcription here, and I have this messenger RNA as a result of transcription. And that messenger RNA codes for the transposase. So I have a ribosome on the messenger RNA translating that messenger RNA, and that's going to lead to production of the transposase. Now what the transposase will do, what we actually need is two copies. We need two transposases here. So these transposases are going to search for the sequences in the TIRs. So essentially they're going to search for and locate the TIRs, and they're going to bind to the TIRs. So here I have the insertion sequence with the transposases bound to the TIRs. And these transposases, they interact with each other. So they have an affinity for each other. So what they're going to do, they're going to bind each other, and that's going to cause this sequence to form some sort of loop here, which I have diagrammed like this. So we have the transposases bound to the TIRs. We have the loop, the insertion sequence loop here. And what these are going to do, and it's something neat about the mechanism, is, you know, when these interact and they pull the DNA in this particular structure here, it induces them to make the cuts that release, the cuts in the DNA that release the insertion sequence from the chromosome. So now we're not going to focus too much on how those initial cuts were made. We will take a look at that in more detail when we look at the Drosophila P element. But for now, we'll assume that the transposase is just going to cut that TIR and that TSD. It's just going to make a blunt end. It's going to cut on both sides of that DNA molecule. So we have a blunt-ended TIR over here on the end of on one end of the IS element, and then we have one of the TSDs over here that's blunt-ended. So I have this thing right here diagrammed down here. And so you can assume the other thing is happening on the other side. So we got these blunt ends right here. And again, they may not be blunt-ended. We're going to not worry about that. Assume they're blunt-ended, and they are going to be fixed by DNA repair, whether that be non-homologous end joining or double strand break by homologous recombination. Some repair process is going to stick these pieces back together. Okay, so that insertion sequence might no longer be there, you know, right? Depending on what DNA repair process was used. But the transposase, okay, it's still stuck. The transposases are still bound to the TIRs, which are bound to the rest of the insertion sequence. Okay, so this can now move to the transposases can now move the insertion sequence to a different part of the genome. So here I have a different part of the genome, and if you take a look at the notes, you're going to see that figure 15.3, which is from our recommended textbook for the course. And what you're going to see is a bunch of different IS elements with their characteristics of, or their preferred target sites. So some transposases from some IS elements prefer target sites of certain sequences. So the one, our hypothetical transposase here, I'm saying I am imagining it prefers this sequence for a target site. So it's looking through the genome and it finds an ATGCA. So it's going to use this to insert the insertion sequence. Okay, so how is it going to do that? So we have two transposases here. So each of these is going to cut the DNA in a specific way on either side of the target site. So it's gonna, one of them is gonna nick right here between this A and this. I have these dots here. I didn't want to put the actual base pairs in here. That would just make it too confusing. So this A and whatever base is represented by this dot, one nick would be put in this top strand, and then the other transposase will put a nick right down here. Let's say downstream of or upstream of this T, depending on how you think about it. So between this T and this dot right here. So it essentially makes a staggered double strand break, right? And so by nicking the DNA in that way, we can get something that looks like this. You know, I didn't put the transposases here, so actually I'm not really sure what the transposases do at this point, if they're still bound here. Perhaps they are. And perhaps this is still some sort of loop here. I'm not really sure about that. But we can see the result of the double strand break, the staggered double strand break, right? Is we have now an ATGCA up top and a TACGT down bottom. Right? So maybe I can diagram it like this. So one break was made here, and another break was made right here. Let's see, is this working out? I think that looks good, right? So now these can get pulled apart. So this can go that way, this right here can go to the right. So ATGCA went left, TACGT went right. And the transposases, you know, probably working with other enzymes, can have this TIR ligated to this point right here, and this TACGT ligated to the TIR right there. And now we have these gaps that need to be filled in. And even before I fill those gaps in, you can see that, you know, probably see that how the target site was duplicated, right? So the target site was TAGCA, the bottom strand was TACGT, right? So we can see the ATGCA went left, the TACGT went right. So now once we fill those gaps in, oh, we're essentially duplicating this sequence. Okay, we can see that the target site has been duplicated. And so here we go, ATGCA, ATGCA. Okay, so how are the gaps filled? Essentially any mechanism in this course where we see a gap in the DNA after ligating something like an insertion sequence in, we see some gaps. How the gap's gonna be filled? Well, a DNA polymerase is going to come in and it's going to fill in the gap. There's going to be a nick, and the nick's going to be sealed by a DNA ligase. So I just didn't diagram all those steps because by now it's probably second nature to all of you that is how gaps are filled and nicks are sealed by DNA ligase. So okay, so we have the insertion sequence. It moved to a new location, and the target site was duplicated, and we still have the TIRs, right, on the ends. So, you know, so when I was first, you know, learning about these types of elements, one of the first questions that, you know, I thought about was, you know, how do transposable elements, class 2 transposable elements that, you know, move to new locations by the cut and paste mechanism increase their copy number? So let's say this is an E. coli chromosome, and this is an IS element right here. And if it moves to a new location, right, it's just going to, you know, be cut out of here and moved to over here. So here is region 1. I'm diagramming how I think, okay, it's going to move over to region 2. You know, how do the numbers increase? Because these elements make up large percentages of our genomes. I think in humans, transposable elements, all the classes together make up about 43% of our genome. Okay, so well, how can elements that move by the cut and paste mechanism increase their copy numbers? And I just want to go over one possible mechanism. I like this one. I think it's a pretty easy, I guess, mechanism to understand. A mechanism to explain how a cut and paste transposable element can move to a new location. So let's go with something like this. Okay, so here is the IS element in region 1. Now let's assume that, okay, it's happy here, just doing whatever it does before it, you know, moves. And then let's say a replication fork moves past that location, so it replicates that part of the chromosome, and now we have, you know, a copy of the IS element on both of the products of replication there, and the replication fork is still moving, so replication is still ongoing. Now, while replication is happening, you know, it's still going on, So let's say at that point, sometime, after this region has been replicated, the transposases lock onto the TIRs and cut out the IS element. That leaves a double strand break that must be repaired by whatever DNA repair processes the cell uses to fix double strand breaks. And so non-homologous end joining could be one, and these could just be stuck back together. And then, okay, there's no more IS element in, this product of replication down here. Keep in mind, replication is still going on. It's still, the replication fork is still moving from left to right. So now we can take region 2, which did not have an IS element, or that particular IS element. It has not been replicated yet. So let's say that replication fork, no replication fork has passed over region 2 yet. Still before the replication fork has passed that region and replicated it, the transposases with the IS element lock on to the target site and integrate the IS element into region 2. That gives us something like that. Okay, now we have the IS element at region 2. And finally, the replication fork makes it to that region, and now region 2 has been copied. So now we have a copy of the IS element in both products of replication, and eventually replication is going to finish. So after all of that, what do we have? Okay, so we have the 2 products of replication. Let me see if I can go back. Good take. So this is what we started with, before DNA replication started. And because of, where the replication forks were relative to the movement of the transposon, we have 2 products of replication. Region 1 and region 2 both have a copy in one product of replication, and the other one has a copy in region 2, but no copy in region 1. And I'm still showing the break here, so we have to assume that the DNA repair processes of the cell are going to fix that break. And if they fix it by let's say non-homologous end joining is used to fix that break, well, that'll be stuck together, and this chromosome, this product of replication, will have only 1 copy, so no net gain here, no net gain in the number of that IS element in this chromosome. But over here we have 2 copies. So that's one possible way a cut and paste transposon can increase its numbers in a genome, even though it actually has to be physically moved from one location to another. Okay, I hope you understood that, and please, as always, email me if you have any questions. In the next video, we will take a look at a specific cut and paste type transposon, the Drosophila P element.